Hi all! So today we're going to talk about nucleic acids. We'll talk a little bit about their function and a little bit about their structure and how those two relate. So we're going to start out with DNA, which is deoxyribonucleic acid. So DNA. Now DNA is pretty much the master copy of instructions. And those instructions are for building proteins. Now, the proteins are the ones that really do all the work in a cell. But the DNA gets a lot of credit because without it, you couldn't make protein. So it's your genetic material. It's super important because it tells you how to make functional proteins, and then the proteins do all the work and give you your traits. So DNA, you may remember, if you're looking at a eukaryotic cell like you and me, the DNA is stored in the nucleus. So eukaryotes they also have it in their mitochondria and chloroplasts, but it's a little extra. If you are a prokaryote, like a bacteria, then your DNA is going to be floating freely in the cytoplasm. So if you're talking about eukaryotes, it's going to be found in the nucleus. If you're talking about prokaryotes, there's no membrane-bound organelle, so it would be found in the cytoplasm. But DNA is universal. No matter what kind of organism you are, prokaryote, eukaryote, bacteria, fungus, plant, all of you are going to use DNA. And you're going to use it pretty much the same way. So what does that mean in like big picture? I can actually take a gene from, say, a human, so like the gene for insulin, which is a super important protein in regulating blood sugar, and I can put it into a bacteria, and that bacteria will not only be able to incorporate that DNA, but it would be able to read it and make the protein. So they use the same language, which is actually pretty cool if you think about it. Now let's look a little bit closer at DNA's structure. So DNA is a polymer made up of nucleotide monomers. So if you remember, a polymer is made up of a long chain of monomers, so poly many, mono one, and the nucleotide is that kind of base unit for DNA and RNA. We're going to look at a DNA nucleotide first, and these are going to be made up of a sugar, attached to a phosphate group, and one of 4 possible nitrogen bases. You can kind of think of this as the up house, like the house and the mailbox and the balloons. That helps you remember the structure. So in DNA, the sugar is going to be deoxyribose. Remember that ose tells you it's a sugar. The phosphate, and then you have one of 4 possible nitrogen bases. You have adenine, thymine, guanine, or cytosine. So usually we just call them A, T, G, and C. So I do want to point one thing out before we move on to the next part. This sugar, deoxyribose, it's a 5-carbon sugar, and so when you draw them this way, anywhere you have these kind of bends, that actually is shorthand, and there's a carbon there. And there's actually one right here. And sometimes we'll number those carbons. So you start at the oxygen, and you go around clockwise. So I'm going to say 1, 2, 3, 4, 5. And this is actually important because when we start talking about which direction that nucleotide is facing, sometimes you'll see them talk about the 3' end, and then other times they'll talk about the 5' end. Well, the 3' end just means that this part is sticking out, or you're talking about this end, and the 5' end is this end up here. So it sounds complicated, but it's just talking about which direction are we facing. All right, so let's dig a little bit deeper into how these monomers get put together to form DNA. So DNA is sometimes referred to as a double helix. And one thing you can pull from that is double, it's double-stranded. So what does that mean? It means that I have 2 chains of nucleotides, and they put themselves together in a particular way. So I'm going to have here's one nucleotide. So there's my sugar, my phosphate, and here's one of the nitrogen bases. It attaches to another sugar, phosphate, nitrogen base. Sugar, phosphate, nitrogen base. And this is just one strand. So where's my other strand? How does that come together? Well, it comes together like this. It's always harder for me to draw it this way. Sorry, that one connects there, that one goes like this. This connects here, and then this goes like this. That's what that's for. All right. So again, I've got my sugar, phosphate, nitrogen base, sugar, phosphate, nitrogen base, sugar, phosphate, nitrogen base. Now, let's say this was an A, this is a C, this is a T. This T is going to base pair with an A. And this C is going to base pair with a G. A, T. And these we say complement each other. So A always base pairs with T, T to A, C to G, G to C. In DNA, it's called the base pairing rules. I'll write that down, don't worry. But I just want to point out a few other things about this particular model. So These lines that you see over here and over here, these are all representing covalent bonds. And in between these two nitrogen bases, those are going to be hydrogen bonds. And you may remember from when we were talking about our biochemistry unit that covalent bonds are stronger bonds and that hydrogen bonds are weaker kind of interactions. And that's actually a good thing because that means that you can open this up to read the information stored in these nitrogen bases relatively easily without kind of destroying the whole molecule. So you may also notice that these are pointing in different directions. So let's go back to that whole 5-3 thing. Well, this is my oxygen. I have 1, 2, 3, 4, 5. So this is my 3' end, and this is my 5' end. And then here, again, if this is my oxygen, 1, 2, 3, 4, 5. Well, okay, this is a 3' end, and this is the 5' end. So if I look at it from 3 to 5, they go in opposite directions. We say that the strands are anti-parallel. Means that strands point in opposite directions. All right, a couple of other things. So we've got our hydrogen bonds. This right here we call the sugar phosphate backbone. And so you have the sugar phosphate backbone on both sides. And then on the inside, kind of facing in towards each other, is where you have your nitrogen bases. And that's actually super important. These nitrogen bases and the order that they're in is where all the information for DNA is stored. So, a couple of things to remember here. You've got your base pairing rules. So, A base pairs with T, C base pairs with G. Again, T goes to A, G goes to C. Now, I used to remember this when I was in high school, that the straight ones, like A and T, base pair together, and the curvy ones, C and G, base pair together. I don't know, it worked for my brain. But then I came up with some other tricks. So, we are the Anderson Trojans, right? So, Anderson Trojans, AT, so CG, chips and guacamole. Whatever trick you use, just make sure that you have some way of remembering. So A always goes to T, C to G, T to A, G to C, if we're dealing with DNA. Now RNA is a different story. So let's just look at RNA. So RNA stands for ribonucleic acid. So RNA. And the RNA is like the working copy of the instructions. So remember, DNA was your master copy that gets stored in the nucleus. You don't want to mess that up because it's like your copy of everything. But you don't always need all of your instructions all of the time. So RNA copies the section that you need to make the protein that you need. And so it's really your working copy. Now, RNA, unlike DNA, is going to be single-stranded. So you only have one chain of nucleotides, but it's still made of nucleotides. But these nucleotides are slightly different. So notice the name ribonucleic acid instead of deoxyribonucleic acid. Well, that's going to be our first difference, is your sugar. It's still a 5-carbon sugar, but in this case it's going to be ribose, so that's R-I-B-O-S-E, ribose, instead of deoxyribose. Let me rewrite that so it's easier to read, R-I-B-O-S-E. Sorry, my handwriting was bad on that. And then you have your phosphate group again. And your nitrogen bases. And here's another place where we have some differences. So you have A again, so adenine, uracil, guanine, or cytosine. So you have one of these 4 nitrogen bases. So here you can see some of those differences. You have uracil instead of thymine, you have ribose instead of deoxyribose, and it's going to be single-stranded. Now, with this uracil, you have a change to your base pairing rules, right? So, base pairing rules for RNA are slightly different. So let's look at this. So if I have DNA has A, T, C, G, RNA is going to base pair differently. So there's no T to go with the A. It will base pair a U to an A. T still goes to A, C still goes to G, G still goes to C. So this is going to be your main difference. So I always remember like AU guys as my AU. But again, anything you can do to remember that that's going to be the difference is going to be useful. Now there's one last thing you really should remember about RNA as opposed to DNA. And it's that you have 3 different types. So there's different types of RNA. So let's look at those 3 types. Or these are the 3 main types. We have mRNA, which stands for messenger. And it does just that. It carries instructions from the DNA in the nucleus out into the cytoplasm. Then we have something called tRNA, and that is your transfer RNA. And that actually carries amino acids to help build the proteins that the messenger RNA is coding for, has instructions for. And then the last one is going to be rRNA, which stands for ribosomal RNA, which, guess what, helps make ribosomes, which actually put together the amino acids using the instructions from the messenger RNA. So ribosomes are actually made up of proteins and rRNA together. So those are your three main types of RNA. So that's it for me. We've talked about the structure of DNA and the structure of RNA and a little bit about their general functions. I would go through and review and see if you can identify three differences between RNA and DNA. And then also see if you can find at least two similarities. It's a good way to kind of test yourself. If you have any questions, we will go over those in class. I hope that was helpful.